Protein analysis is vital for understanding cellular processes and diseases. This video covers protein extraction, purification, advanced techniques like multiplex Western blotting and co-immunoprecipitation, data analysis, troubleshooting, post-translational modifications, and imaging in protein research. Perfect for medical students in molecular biology. Proteins are made up of amino acids, linked together in a specific sequence. This sequence determines the protein's shape and function. Protein structure is important for understanding their functions in the body, such as enzyme activity or DNA interactions. Problems with protein folding can lead to diseases. To analyze proteins, they must be extracted and purified from biological samples. Protein extraction methods vary depending on the source material, ranging from cellular lysates to subcellular fractions. Common techniques include cell lysis, centrifugation to remove cellular debris, and isolation of desired protein fractions. Following extraction, protein purification is essential to obtain highly pure proteins for downstream applications. Techniques such as chromatography, including ion exchange, size exclusion, and affinity chromatography, aid in separating proteins based on their properties such as charge, size, or affinity for specific ligands. Gel electrophoresis is a technique for separating proteins based on their size. Polyacrylamide gel electrophoresis, PAGE, and sodium dodecyl sulfate polyacrylamide gel electrophoresis, SDS-PAGE, are commonly used methods. SDS-PAGE helps to denature proteins and ensure uniform charge to mass ratios. Staining with dyes like Kumasi brilliant blue or silver stains allows visualization of separated proteins, and densitometry software can be used to quantify protein bands. This technique is useful for assessing protein purity, estimating molecular weight, and studying post-translational modifications. Once proteins are separated by gel electrophoresis, they need to be transferred onto a solid support membrane. This process, known as protein transfer or blotting, facilitates downstream protein detection and analysis. Different transfer methods, such as wet electroblotting and semi-dry blotting, are employed based on the experimental requirements. Transferring proteins onto nitrocellulose or polyvinylidene fluoride, PVDF, membranes allows for subsequent antibody-based detection methods, such as immunoblotting or immunostaining. After protein transfer, the membrane is subjected to a blocking step to prevent nonspecific binding of antibodies to the membrane surface. Common blocking agents include bovine serum albumin, BSA, or non-fat dry milk, which saturate unoccupied binding sites on the membrane. Following blocking, primary antibodies specific to the protein of interest are incubated with the membrane. Primary antibodies bind to their target proteins with high specificity, allowing for subsequent detection and analysis of the protein. Scientists use primary antibodies to detect specific target proteins. These antibodies can be polyclonal or monoclonal, with polyclonal antibodies offering a mixture of antibodies targeting various parts of the protein and monoclonal antibodies providing high specificity by targeting a single part. Different detection methods, such as ELISA, chemiluminescence, and fluorescence-based techniques, allow for the visualization and measurement of protein bands or spots on the blotting membranes. Once protein bands are visualized, appropriate software tools are employed to analyze and interpret data obtained from Western blotting experiments. Densitometry software aids in quantifying protein bands, allowing for comparisons across different samples or experimental conditions. Software packages further facilitate statistical analysis and graphical representation of the data. During protein analysis, various technical issues may arise, hindering experimental progress. Troubleshooting skills are essential to identify and resolve these problems. They include optimization of protein extraction, purification procedures, gel electrophoresis parameters, antibody concentrations, and imaging conditions. Understanding common pitfalls and troubleshooting strategies is crucial for obtaining accurate and reliable results. Proteins undergo post-translational modifications, PTMs, that significantly influence their structure, function, and stability. Phosphorylation, acetylation, glycosylation, and ubiquitination are examples of PTMs that regulate protein activity, localization, and interactions with other molecules. Identification and analysis of PTMs require specialized techniques, such as mass spectrometry-based proteomics approaches or specific antibody-based assays. Studying PTMs provides valuable insight into cellular signaling pathways and disease mechanisms. Traditional Western blotting allows detection of a single protein at a time. However, advancements in antibody labeling techniques and imaging systems have enabled the simultaneous detection of multiple proteins in a single blot. Multiplex Western blotting methods provide enhanced experimental efficiency and reduce sample consumption. To understand cellular processes comprehensively, it is crucial to study protein-protein interactions. Co-immunoprecipitation, co-IP, is a widely used technique that allows the selective isolation of a target protein along with its interacting partners from a complex mixture. Co-IP involves incubating a cell lysate with an antibody specific to the target protein, followed by immunoprecipitation of the antibody-protein complex. The precipitated proteins, including the target protein and its interacting partners, can be subsequently analyzed by Western blotting or mass spectrometry. Imaging and documentation of Western blots are crucial for preserving experimental results and allowing scrutiny by others. Chemiluminescence, fluorescence, or infrared imaging systems are commonly used for protein detection, depending on the detection method employed. Accurate documentation includes annotation, proper file naming, and storage in the appropriate file formats. Ensuring reproducibility and traceability is essential for future reference or publication. By gaining a deep understanding of protein analysis, medical students can contribute to advancements in molecular biology and medical research. This script covers protein structure, advanced techniques, data analysis, troubleshooting, and imaging, providing a comprehensive overview for effective protein analysis.